Hello, welcome to Biology, the scientific study of life. Gene pools and allele frequencies. Before continuing our discussion of population genetics, we need to define what we mean by a population. A population is a localized group of individuals that are capable of interbreeding and producing fertile offspring. Populations of the same species may be isolated from one another, thus exchanging genetic material only rarely. Such isolation is common for populations confined to different, widely separated islands or lakes, but not all populations are isolated, nor must they have sharp boundaries. Still, individuals near the population center are more likely to breed with members of their own population than other populations, and those on average are more closely related to one another than to members of other populations. The aggregate of genes in a population at any one time is called the population's gene pool. It consists of all alleles at all gene loci in all individuals of the population. If only one allele exists at a particular locus in a population, that allele is said to be fixed in the gene pool, and all individuals are homozygous for that allele. Recall that homozygous individuals have two identical alleles for a given locus, while heterozygous individuals have two different alleles at the locus. But if there are two or more alleles for a particular locus in a population, individuals may be either homozygous or heterozygous. Each allele has a frequency proportion in the population. For example, imagine a population of 500 white flower plants with two alleles, S to the power R and S to the power W. At the locus that codes for flower pigment, plants homozygous for the S to the power R allele, S to the power R, S to the power R, produce red pigments and have red flowers. Plant homozygous for the S to the power W allele, S to the power W, S to the power W, produce no red pigments and have white flowers, and heterozygotes S to the power R, S to the power W, produce some red pigments and have pink flowers. In our population, there are 320 plants with red flowers, 160 with pink flowers, and 20 with white flowers. These alleles show incomplete dominance because these are diploid organisms. There are a total of 1,000 copies of genes for flower color in the population of 500 individuals. That C to the power R allele accounts for 800 of these genes, which means 320 by 2 will equal 640 for S to the power R, S to the power R plants, plus 160 by 1 equal 164 S to the power R, S to the power W. Plants. When there are two alleles at a particular locus, the convention is to use P to represent the frequency of one allele and Q to represent the frequency of the other allele. Thus, P, the frequency of the S to the power R allele in the gene pool of the population is 800 equal 1000 equal 0.8 equal 80%, and because there are only two allelic forms of this gene, then the frequency of the C to the power W allele represented by Q must be 0.2 or 20%. At loci that have more than 2 alleles, the sum of all allele frequencies must still equal 1, which means 100%. We can easily measure genetic variability at this flower color locus because each genotype has a distinct phenotype, although many loci in gene pools have more than one allele. This variability is usually less easy to quantify than in our flower example, because one allele may be completely dominant or the alleles may not have obvious effects on phenotypes. The Hard-Weinberg theorem. In a moment, we will explore how genes and allele frequencies can change over time, resulting in evolutionary change. But first, to provide a benchmark against which to measure such changes, we will investigate the properties of gene pools that are not evolving. Such gene pools are described by the Hardy-Weinberg theorem, named for the two scientists who independently derived the principle in 1908. The theorem states that the frequencies of alleles and genotypes in a population's gene pool remain constant from generation to generation, provided that only Mendelian segregation and recombination of alleles are at work. Preservation of allele frequencies. The Hardy-Weinberg theorem describes how Mendelian inheritance preserves genetic variation from one generation to the next in populations that are not evolving. But it does more than this. We will see that the theorem lays the groundwork for understanding long-term evolutionary changes that Darwin, lacking knowledge of genetics, could not have envisioned. The preservation of genetic variation provides the opportunity for natural selection to act over many generations. Let's apply the Hard-Weinberg theorem to our hypothetical wildflower population. Recall that 80%, which means equal 0.8 or 8 part of 10 of the flower color alleles in the gene pool are C to the power R, and 20%, which equal 0.2, are C to the power W. Because each gamete produced by the flowers is haploid, it carries only one allele for flower color. The chance that a gamete will carry a C to the power R allele is 0.8, and the chance that it will carry a C to the power W allele is 0.2. The allele frequencies in all the gametes produced by the population will be the same as the original population, provided that gametes are contributed to the next generation at random. Allele frequencies will be unchanged. Notice that Mendelian inheritance preserves genetic variation from one generation to the next. Notice that generation 1, generation 2, notice the percent also 50%, and notice that generation 3 and 4, notice also okay, unchanged. The allele frequencies will be unchanged. Notice that. Okay, thanks.